Alrighty, hello folks. Our guest today is Richard. How do you pronounce your last name?
Richard Fuisz, like a fusion protein.
Andy, on brand. Richard Fuisz, CEO and co-founder of Nonfiction Laboratories, a biotechnology company developing magnetically controlled therapeutics. Nonfiction is building a platform of engineered proteins, including Magloff2 and mag bodies that respond to weak magnetic fields, with potential applications ranging from targeted cancer therapies to programmable biologics. Richard studied bioengineering at Stanford and has spent his career at the intersection of biotechnology, company building, and frontier science, including founding DNA synthesis company Templa Nucleics and contributing to organizations like Arcadia Science and Future House. Today, he's helping translate discoveries in magnetic biology into a therapeutic platform. Richard, if you would like to go ahead and also introduce yourself, if I missed anything, that would be great.
Yeah, I think that's great. So yeah, I started nonfiction about 18 months ago with Maria Ingaramo and a bunch of almost Andy York, who was also quite on Twitter a bunch. They had begun this work. I really also want to, although they're not here today, I really want to highlight some of what they've done as well. So Andy and Maria were over at a place called Calico Labs for a couple of years, actually maybe like 7 years or so. Uh, they were sort of a new technique development around mostly imaging and got kind of the crazy idea to just start placing magnets uh, next to cells and see if they could get an effect. And turns out that they did get an effect, and that effect initially was super small. But they kept at it, and they started doing directed evolution to make it bigger. I can tell sort of the full story a little bit later. Um, but yeah, they did some really amazing work, and I got to know them through a couple other attempts to build sort of inventor Shangri-La type things. And so once we started to see those effects and they started getting big, then we decided like, yeah, let's do it. So yeah, that's I think you covered me super well. Um, yeah, it's great.
Amazing. Do you want to tell us a little bit about the work you're doing with Nonfiction Labs?
Yeah, for sure. So Maria and Andy discovered this fluorescent effect. So basically what they saw is there's a protein, some proteins are I think called fluorescent. What that means basically is just they have a particular color to them. So you hit them with one wavelength of light and another one pops back up. And they saw that that color of those proteins was changing as you bring a magnet to them. And initially that brightness was like a really subtle thing. It was like, you know, 0.5% different. Like you'd have to do a bunch of like statistics to kind of suss that out. Um, but by doing directed evolution, the effect got bigger and bigger and bigger and bigger. And it got to the point where, under the right conditions, you can get about a 300% change, so about threefold change in brightness. That's the kind of thing you can just see with your eye. You don't need statistics to like notice that. And so that was the really exciting sort of initial result that they had. Um, uh, they published that stuff that was out sort of in the open, and then that kind of got me getting sort of where I got really excited about this was saying, okay, fluorescence is really cool, changing color is really cool, but what else in biology can we actuate? So there's a field called optogenetics where people have basically taken every type of protein, every type of enzyme that you could think of or a biologist could think of, and can have it controlled with light. So they apply light and it turns on or it turns off. Um, and it turns out that the domains that are used in optogenetics, these like the ways they make that work, is they take two proteins and they fuse them together, and then you get a light controllable protein. But those light controllable proteins are very similar to our magnetically controllable proteins. So we got kind of the question of, okay, you have this huge field of like 15 years of amazing work with light. Maybe we could make that controllable with magnets, not light. And the reason why that was so exciting is basically just that magnetic fields penetrate really deeply into the body. So we had this opportunity to do sort of remote controllable therapeutics. There's a lot of other ways people have thought about remote control, but a lot of them have had challenges for a long time. But if you could control something with magnets, it would be really, really amazing. So that's really exciting. So that's sort of what we set out to do. We started the company about 9 months ago. We got our first glimmers of magnetically controllable antibodies, which is very exciting. So we've been sort of chasing that since And along the way, we've made a ton of other enzymes and all sorts of other proteins that again are all sort of inspired by the optogenetic work that came out of Ed Boyden's lab and Carl Desseroth. Um, uh, and really like just sort of adapting those to be magnetic tools. So that's like mostly what we spend our time on. We have a lab in San Francisco. We have a lab down in Argentina as well. Actually, the majority of the staffing is now in Argentina these days. Fantastic team down there. And then we also like are part of this much broader community, which includes, of course, quantum biology DAO and QBI, and then a ton of other academics at Stanford and Harvard and Oxford, and then ton of new places now too. Some amazing collaborations, which we're really excited to chat about. There's some great work that came out of Northwestern recently, some stuff from UC Irvine as well. So it's just been so exciting to see these proteins that we've been making get into the hands of people, and like really start to shape some of how people can think about problems. And some of the most interesting work right now is just sort of asking like, how the heck do these proteins work? Because like that's an interesting question. So yeah.
Yeah, go for it.
Yeah, Kira, do you want me? Yeah, you want me to jump in?
Yeah.
Okay. Anyway, and so for people who don't know, I'm Jeff Anders, co-founder at the Quantum Biology DAO. Um, Great to be chatting with you, Richard. Spoken before, of course.
Yeah.
I actually wanted to pick up on the question that just came up. Yeah, so why do you think why have people not looked into magnetism as a way of
Yeah.
actuating inside the human body? And
Yeah.
do you have any ideas about what like it's I guess why have people not looked into it and how does it actually work?
So some people, the very sciencey answer is always like, well, it depends, and technically people have, and so that's true. So people have tried to look and say, well, maybe magnetic fields can control some things. And we know that there are some, there are effects that many different things seem to show when you apply a
Mhm.
magnetic field to them. Magnetic fields do affect some like oxygen species, and there's some things like that. But typically these effects have been like very small. So you have to be very good at measurement and very good at statistics to see them.
Mhm.
And the thing that Maria broke out, which is so exceptional, was like starting to apply directed evolution to this problem, getting
Right.
that effect much larger. And then you're not quibbling about like, well, was it 0.5% or was it zero? Because there's a lot of things that can cause 0.5% effects, like random statistical chance, or like the device is, we've run into all these sorts of problems of like the magnet is tugging on the lens of a camera, or the magnet is triggering a signal to fire through a wire that we didn't think was supposed to fire. So, the timing is getting messed up. There's a lot of like goofy
Right.
little things. But when you can get it up to 300%, then those things sort of go away. And you're like, okay, there are effects. So that was,
Mhm.
I think, that was a new thing. Whereas when people have looked in the past, kind of biologists just like to steal from uh, existing, like they like to steal something that like already exists in nature.
Right.
So there's a lot of plants that it's very important for a plant to know where light is. So it's really nice if a protein can do that.
Mhm.
There's not as much sort of historical reasons why an animal would need to strongly respond to magnetic fields. And to the extent that, may be true at all, which is of course still being like, it's a very active area of quantum biology. The fields that are typically experienced by life until the last like 50, 100 years were orders of magnitude weaker than a fridge
Mhm.
magnet, right?
Mhm.
So when we come in and we're applying fridge magnet and stronger
Right.
fields, that's just totally like foreign, exotic stimulus for biology to experience. Um, so I think basically there's a lack of things to steal. Because there's a lack of things to steal, all the effects were quite small.
Mhm.
When effects are small, people get a little bit sketched out. Um, and then also when you're dealing with small effects, like then you have to ask the question of, well, there's a bunch of some of these small effects might actually matter, right? So if you're
Right.
continuously exposing, if you're shifting something about ROS or something like that, you might have an effect. I would say the other final bit too is like the general assumption is that at least static magnetic fields, alternating magnetic fields are a slightly different story, but certainly for static magnetic fields, there's really not any safety stuff that has usually come up. Like MRI technicians, people who are exposed to very strong magnetic fields don't have dramatically different health outcomes, or usually even different health outcomes sort of at all that are noticeable. So the idea is like it just makes it a little bit hard to imagine that like static magnetic fields are causing uh, are going to be
Okay.
a hugely explosive thing. So I think that's kind of like the
I see.
there's
That
probably
makes sense.
less oscillation.
I have seen some things about people having negative reactions if they're in MRIs for a while,
Sure. Yeah.
but yeah, I guess it makes sense that if the effects of magnetic fields are subtle and then you start bathing people in electromagnetic radiation of different types
Yeah.
and we seem to be fine, and the magnetic fields are messing up your electronics, and so you're constantly trying to, if it's more a nuisance than anything else, then you wouldn't expect there was the sort of thing that could be harnessed.
I think that's right. And I would say, so just a small caveat on some of the MRI stuff. My understanding, the stuff where you see that most commonly, so people will get like optical flashes and like really strong MRIs or tingling or sort of neuronal firing, stuff like that. That's often caused by movement within a static magnetic field. Because if you're moving in a static field, now you kind of have an alternating field. So you could get current generation and stuff like that. So,
Okay, that's
um,
interesting.
yeah, so there's some nuance. There's some nuance with like static magnetic fields are really purely static, right?
Okay, but so
But
you
the,
could, I guess you could imagine a sort of imagine that we're in the world where biology hasn't learned to do very large things with magnetic fields, but that doesn't mean biology can't. It's just
yeah,
we didn't. And then if you directly evolve things outside of the domain that we normally work in, then you find that actually you can get really large effects from magnetic fields. Is that sort of the idea?
yeah, I think that's right. I mean, maybe to say it more succinctly than I'd said it before, like the small subset the small subset of biology that we have studied, we haven't seen large magnetic field effects in naturally occurring proteins. But the fact that you can evolve a strong effect like implies that like this is possible. Like biology
Yeah.
can do this, which is a very interesting conclusion, I think, from sort of just the scientific like ramifications of that, right? And like maybe why it's worth looking more for these effects.
Yeah, I mean, it sounds really exciting. How have people responded to the fact of maglev 2? Like how or mag
Yeah.
is it help me straighten me out here. So is maglev 2?
Oh
What?
God, the naming is all a mess. Yeah. Um, so there's maglev 2. Sometimes we'll refer to there's a class of proteins called love domains. That's LOV.
Okay.
Uh, and those love domains are quite common across many different types of biology. Like they're very similar things. There's a related family of cryptochromes,
Hmm.
which are like the eyes of mammals and various other sort of animals. Um, love domains are all sort of they look kind of similar to each other. They're usually a flavoprotein, so they have a flavin kind of
Hmm.
embedded in them. Something like 1 to 2% of all proteins, I think, are flavoproteins. So
Hmm.
flavoproteins generally not super uncommon. Love domains quite common across many species. There's a particular protein domain, which is what Maria and Andy started with, which comes from wheat. It's called ASLOV2.
Hmm.
Uh, but then there's tons of other species have their own version
Right.
of ASLOV2, and those all have some slightly different properties. Um, but it does seem like uh, and the fluorescent proteins as well, which can also have like their own separate versions of these effects.
Hmm.
It can be engineered and can be enhanced. Like GFP can have a mild magnetoresponsive effect, which is pretty crazy.
Hmm.
Um, but yeah, so when we talk about maglev, usually we're talking sort of in the broadest sense, we're usually talking just about like love domain or love domain like proteins that can do this
Right.
type of thing. Um, and sometimes we're also talking about the original maglev, which is like a particular sequence derived from this wheat protein.
Okay. So is aslov 2 to maglev 2? Is that the order that it happened in?
That's what it should be. Yes, that's what we should try to keep it straight and have it be. Yeah.
Okay, okay.
Yeah.
But so then after you guys got to Maglev and Maglev 2, how did
Yeah.
people respond? I think this sounds like a
Oh,
big deal.
Yeah, people were super excited. Yeah. I mean, so there's been two, well, first off, there's been two Nature papers in the last 6 months on that are sort of maglev related, which has been great.
Okay.
So like the traditional academic like publishing ecosystem, I think is like taking off with it, which is awesome. Uh, there's a ton of outreach. I mean, I think the initial response, and this is just what I've heard from Maria and Andy, it was like uh, people were a little bit suspicious because magnetogenetics has been a challenging field, and so people are like, "Oh, I want to replicate it first in my own lab."
Okay, sure.
And then the fact that it works with a strong enough effect means that even if you don't get everything perfectly right, you can still see a signal without too much fiddling. So the fact that
So
there was
people replicated it.
exactly a lot of people
Okay.
replicated it, uh, and the fact that the hello world is quite easy. It's like you have a regular microscope in your lab, you buy a magnet, you buy a popsicle stick, you tape them together, and then you wave it. And like Maria was fielding so many questions from people being like, well, what sort of what servo motor do I need? And like, what's the, it's like, no, like all the initial stuff is just off of a popsicle stick.
Right. Okay.
There's funny photos of that rig. But so people started replicating it. I think the first big question people had was like, does this have all the properties that love domains have, like standard as love? That was an early exciting question.
Yeah.
Uh, One of the big initial ones also was like, uh, does it experience a thing called magnetic resonance, which is really the focus of these two Nature papers that
Okay.
came out. There's also a third paper from Peter Moore's lab, which has a similar bent on this as well, which came out of the same sort of work.
And so,
Uh,
so maybe you should
right.
explain magnetic resonance
For
for,
sure.
yeah.
Yeah. So what magnetic resonance is basically this is sort of the way like an MRI machine works. If I have a magnetic field gradient that's super high here, super low, high on the left, low on the right. And then I apply a radio frequency, so like a microwave or some sort of
Mhm.
like some emitter like that. Um, there's a particular field strength and a particular frequency where those two are going to overlap, and we'll get a blip of increased activity right there.
Okay.
And this is exciting because this is a way to get like spatial precision that can be really, really tight. Like this is why an MRI machine works well, as you
Uh,
can have this nice resonance peak. So they measured it out mostly through a method called ODMR, which is basically it's just these proteins glow. They have this fluorescence to them. That fluorescence effect changes with the magnet, and they could see that you have a little spike of that fluorescence change right
okay.
at the peak. And then if you sweep the frequency up or down, or you sweep the magnetic field strength up or down, you could just move that peak around. And the bit that's exciting about, so what I'm just describing there is like a 1D plane. So
Mhm.
we're like 1D axis will have sort of a plane which is established there. But imagine if I kind of wobble that plane around, but I keep one point in the middle steady.
Mhm.
Now I've created like a voxel.
Mhm.
So now I have like a really precise way to map out and kind of etch exactly the shape I want of where I want the activation to be because I
Mhm.
can kind of sweep that point around, and there's some
Mhm.
amount of delay which will sort of lock stuff in. Um, so that was two exciting things, and then one of the groups done at Stanford, Sean Bird and Mark Kasovich and the Hall, they did a really cool thing where they just put it into C. elegans, which is a worm.
Yeah.
So it showed that in fact it also works in worms.
Okay.
This is an important sort of thing to figure out because like sometimes you learn that proteins work in one species but don't work in another. So far, it's worked in every species or cell type that we've tried. Um, so that's good news. Not dependent on like some exotic cofactor or something like that.
Okay. So in other words, it works in a test tube and it works inside cells so far.
Yeah, yeah, and it works in no cells at all, which is also exciting. So like you can just have the protein, raw protein, that'll work, which is fun.
Huh.
Um,
That's
yeah.
fascinating. Okay. And you've mentioned, I think, the question of how it works physically. How much do you guys know about how it works physically?
We're at the point where we have some glimmers of how it works,
Okay.
but things are still confusing. I mean, and this is an area where I think a lot of great folks, including QBI, is doing some really cool stuff. Um, so high level, you have this flavin, and
Mhm.
I'm going to be very high level in how I explain it because Maria will
Sure.
probably be able to explain it much better. But you have a flavin that's trapped inside the protein.
Mhm.
Uh, in a typical love domain, what the cartoon people would say is that flavin gets photoexcited the electron you get this excited electron, and then it forms a bond with this nearby cysteine, which is a type of amino acid. It's like a little spot on the protein. So the flavin fuses onto the protein, and then electrons shift around, and then the protein is shaped kind of like a head and a jaw,
Mm-hmm.
and then that jaw just like shoots off of the body, and it goes from being this highly ordered kind of U shape to this like
Mm-hmm.
super messy spaghetti shape.
Mm-hmm.
And like if you're fusing that into another protein, you can just mess up the other protein because you've like,
Mm-hmm.
you know, it was once ordered and now it's like all over the place and kind of
Mm-hmm.
opposing itself. Um, with maglev, it's a little bit more confusing as to what's happening because one of the first mutations that showed up in the maglev engineering process was to remove that cysteine.
Mm-hmm.
Um, so it's a little bit unclear exactly what's happening, but the high level answer is probably still something to do with some electrons are getting so flavin gets excited, some electrons are getting sucked off or sent around to places where they don't belong, and then the protein is either the love domain is changing shape or it's just rearranging charges in such a way that it's pulling charge or giving charge to the protein that it's fused to. Um, and then the question is like, where does the magnet get involved? And the answer on that is like a weird quantum thing, which I'm sure the folks at QBI could explain much better than me. Uh, and they have some nice papers that go into this and some nice modeling that they've done. Um, but the high level there is like uh, so that electron gets excited. There's two different states it's balancing between, and then the magnetic field is basically inhibiting some of that switching between these two states that the electron can take. So the magnet isn't actually like it's not applying force because there's no energy delivered by the magnetic field. What it's doing is it's like a control signal um, that's just sort of it's a valve. It's not a pump.
Right.
Um, yeah.
Yeah, it's interesting. I know that QBI has done, this is the Quantum Biology Institute, has done their own magnetofluorescence studies, and I think that
Yeah.
they find that when they apply magnetic fields, the fluorescence dims.
That's right.
Okay.
Yeah.
Whereas when you guys are applying it, the fluorescence is increasing. Is that right? Or is it dimming?
No, we get dimming too, actually. Yeah, yeah. Dimming is the
Okay.
typical easier effect. So kind of part of what maybe what's happening there is like you have two different states of the protein. Uh, if everything is going according to plan in the normal way, then it's just fluorescing back to the ground state. So the light is given off because
Yeah.
it's de-energizing.
Hmm.
And something is happening with the magnet it's applied where it's stopping that usual process from happening and it's
Yeah.
encouraging some other thing to happen where maybe it's like turning into some disordered state and then it's getting stuck in that way and then it's
Yeah,
not
I
getting
think
down as
this is
fast.
where I think the quantum mechanism comes in. The typical explanation is that the
Yeah,
magnetic field causes the excited electron to change its spin state in a way
yeah,
that makes it harder for it to decay back down to the ground state.
yeah, exactly. Yeah,
Yeah, okay. So
yeah,
that's fascinating. And then the fact, I keep thinking about the fact that we don't see these large effects from magnetic fields in nature.
yeah.
And
Weird.
my first thought was, well, maybe this is just an unexplored evolutionary path where we like land on an alien planet and we find that everything is really, we're strongly responsive to magnetic fields, and it's just not the way it turned out on Earth. That was like one thought. But
Yeah.
then the second thought was that we're here talking, I'm thinking about this. What if the magnetic fields are actually doing something, but usually what that is more internal? So that it's like you could imagine that, I mean, this analogy, I'm speculating here, but there's a cool phenomenon. It's like you could imagine somebody who'd only encountered electricity in the form of wires in a modern house, and they're like, electricity, all it doesn't do anything. Like there's no, and then they come across a frayed wire and the frayed
Yeah.
wire sparks them. And they're like,
Oh,
okay, wow, there's a real effect here.
there's an
We know
effect.
electricity doesn't do anything regularly in the house, except that when you get one of these frayed wires, then we can see it has a large effect. Does that analogy make sense?
Yeah, I think that makes some sense. The one thing I would say, so there's a question of, well, like because there's things that things evolve for, and then there's things that nobody evolves for, but like are incidentally true. Like
Sure.
it's convenient that like, whatever, inflammation responds to Advil, but obviously we didn't evolve to do that.
Sure. Yeah.
Um, I think the bit that is maybe particularly challenging about accidentally evolving magnetoresponse that's super strong, there's maybe two aspects of it. One is
Hmm.
like maybe it's just less efficient, like kind of to have a good magnetic effect, things need to be a little bit balanced on like a razor wire. Like
Hmm.
it needs to be, it's not a very strong difference. And so things need to be kind of calibrated that they could flow to this path or this path without too
I
much.
see.
So,
Okay.
so a little bit like evolution tends to not like evolution prefers like the
Yeah,
opposite
of
of
course.
not
Yeah.
having.
Yeah.
So that's maybe one thing. And then I think the other aspect is like, um, because it's not an energy source. So humans maybe haven't evolved to like respond to electrical shocks, but electrical shocks, because they apply energy, they mess up the ion gradients and all the stuff of like nerve cells. And and the body's very sensitive to like the relative positionings of ions inside, outside the cell, or that sort of stuff. So
Wait,
like,
don't magnetic fields apply energy? Like if you have a magnet and a compass needle, and like you imagine you have a stationary compass needle and it causes
oh,
it to move?
um, it's a good. So it's like potential energy. It's like a hill doesn't apply
Hmm.
energy to a ball that's sitting at the top of the hill,
Hmm.
but but there's a way to like, like sometimes you can get some kinetic energy as it chases a lower potential energy state. Um,
Okay, so it's like things could be out of alignment with magnetic fields, and that could be a type of potential energy.
Yeah, so the two ways that magnetic fields can cause that effect, one is they can, if it's flipping on and off very fast, you can get
Hmm.
electrical current that gets generated.
Hmm.
The other thing is like just metal objects can get tugged by
Hmm.
magnets. But there's not
Hmm.
enough metal in the body to like really get good tugging going on in most cases.
There,
Um,
there are surprising concentrations of magnetic nanoparticles, but nobody knows really what they do. So I think,
yeah,
yeah,
yeah, there's exactly there's yeah, so there's I mean like in ferritin, like there's like there's some of these things, but it's a little tricky to yeah.
yeah,
Uh,
okay. Well, so this
exactly.
is there's I think this is a great area where there's a whole bunch of unanswered scientific questions. Let's go
For
back up
sure.
to the level of like
Yeah.
practicality. The
Yeah.
so why are magnetic fields exciting from your perspective, like as compared to like
Yeah.
using light or using ultrasound is interesting. You've mentioned things
Yeah.
being able to penetrate tissue. Ultrasound, I think, is pretty good in that regard. But yeah,
Yeah.
why is magnetism interesting?
Yeah. So light. So okay. So superficially, like light should be great because like we have this whole massive data set of like great tools that work for light. Like it's just like a researcher's wet dream. It's fantastic. Like you just put it on
But
a
we're
plate.
we're not we're not transparent. That's the problem, right?
Exactly. Yes. So then the problem is that you go to, and then you look at, well, where do we actually want to apply biology? And that's like, well, we want to apply it to humans. We want to apply it to like, like mice.
Right,
And like none of these things are very transparent, right? Like maybe you can get a little bit of stuff in zebrafish,
right.
but like you're really pushing it really, really fast because yeah, stuff is opaque. Like the classic, I'm gonna steal this from Andy. He always does this. Like the thumb, you can get a little bit of red light through, but the
Yeah,
palm, you can't get anything through at all.
right.
And
Yep.
so like, if you use that with your like phone light, um, yeah. So opto ends up kind of just like dead in the water for anything more than like a centimeter generously.
Hmm.
But in practice, from a researcher standpoint, it's like a millimeter is usually what people will cite as
Okay.
very good
Okay.
control and depth. So so yeah, so light is kind of out, unless super superficial. Um, Ultrasound is better in that you can go deeper.
Mhm.
Ultrasound has a couple sort of distinct problems that are a little bit challenging. The first challenge with ultrasound is um, that it struggles to get through bone and air.
Okay.
So things where the density gradient changes really fast,
Mhm.
and that's hard because like you have ribs and you have air in your gut and air in your lungs,
Mhm.
and like if you just try to chart a course to wherever you might want to send the ultrasound, just whenever you in the abstract it sounds good, but when you think about any specific case, usually you have to get through bone or get through gas. to get there.
Okay.
So that's the second big challenge with ultrasound. There are fancy like phased array transducers that maybe are making this a little bit easier, but it's still a really challenging like scientific problem to do.
Does
Um,
this mean that Midjourney's full body ultrasound is not that exciting for people who've not heard about this Midjourney, the image generation company has gone into ultrasound?
I think it's pretty rad just because I think it's cool. It's in like his like basement. I think that's freaking rad. So yeah, no, I think it's so. I mean, diagnostic ultrasound, like there's some tricks, additional tricks you can play. If you're
Okay.
willing to put them in a tub of water and have a ring of transducers all the way around them,
And this
you
is
can
what they
start
did. They basically have this
exactly.
huge like tank you go into, right? Yeah.
Yeah, I think that'll be I think their probable long-term advantages there are like the fact that it's going to be so cheap and kind of fun to get a scan.
Mhm.
That means you might get it frequently, and then you can look at changes over time.
Okay.
And so there are going to be some advantages to that, but they have to deal with this bone problem as well.
Right.
And I'm sure if you looked at a super obese patient in their study where you have a lot of different masses and density to
Right.
deal with, those scans will look harder to understand.
Okay.
Um, the other challenge with ultrasound is also just like so if you have an ultrasound hitting a given area, you will cause some heating and you will cause some damage. And those are small amounts of damage, but if you were to have a transducer like on 24/7, that might
Right.
not work.
Right.
So you're probably not in many cases talking about constant continuous excitation.
Okay.
Um, and then the other final challenge I think that is hard from all this stuff too is like and maybe this is the most important one, and sometimes we think about this a lot. It's like ultrasound still and sonogenetics, maybe broadly, does not have their equivalent of a really good love domain.
Mhm.
Um, so what that means in practice is like they don't have really good reversibility. So
Okay.
drugs and proteins all diffuse around the body. So if you look in a given area, so let's say I use ultrasound to deliver or activate a drug at a certain area, within 24 hours, that drug will be a lot of other places that it didn't start at.
Right.
Uh, and it's really nice if when it leaves the area, it turns off, and it comes back into the area, it turns back on again.
Okay.
And
So I see the
you
idea.
can't catch that with ultrasound. Yeah, so that's maybe
So
the yeah,
okay, so if I'm getting it, the idea is that this is sort of like the magnetic resonance, where you can use magnetic fields to pick a point in the person's body where something will switch on. And so then if you have things that only switch on in response to magnetic fields, then then you can turn on antibodies here and have them to not be on elsewhere.
that's right. That's right. Yeah. So I
Okay.
mean, the magic of magnetogenetics is like the reversibility and the switching
Hmm.
characteristics of that. So it means we can apply a magnetic field to your arm. Great. We can have the drug active in your arm. If the drug floats out of the arm, the drug turns off. If the drug floats back into the arm, it turns back on again. And like a magnet can be a static device that you can wear at home, and like it's just a lot easier than like sitting in a bath of transducers like all day long, uh, which is going to be a harder.
That's a harder sell. It's more expensive. It's,
Yeah, it
you're
makes the
covered in
med
more goop.
yeah, it makes the med spa a little less fun if you have to be there all day long, right?
Yeah.
But
Okay.
um, yeah, so I think that's kind of those are the big differences. Uh,
Okay.
yeah,
So then you guys have been trying to build a company around this, and I'm interested in how do these things compare? It's like you have science on one side and you
yeah,
have commercialization on the other.
yeah. So
Similar skill sets, totally different. One's easy, the other's hard. Both are hard. Both are easy.
well, the stuff. I haven't been spending as much time, not really any time at the bench in the last 9 months. So the science bit is very easy from my experience of it.
Right. Okay. Sure.
Um, but it's not as easy for the people who actually are at the bench. I think we've been surprised. Like sometimes I think we've been surprised sometimes as we've learned which things are easy and which things are hard to do scientifically.
Hmm.
And I think that like there's a really important aspect when you're doing something that's so new of just trying to get really good at cheaply experimenting and trying something quickly. And by
Hmm.
cheaply, I usually just mean like try it and then stop doing it if it's not,
Right.
like how do you do like a 6 month and sometimes it will take like 6, 9, who knows, it can
Hmm.
take a while until something starts working well, but how do you kind of cover the
Hmm.
landscape quite well? Because sometimes you find that like at the very beginning of something, it works really fast.
Hmm.
Um, whereas other things are like these slogs really fast. Um, so the science is hard because it's like new, but we're figuring things out, and like
Hmm.
it's kind of our area. Like we've got great people working on the direct
Right.
evolution of this.
Right.
We've got this around that, which is good. The business part, the hardest thing I think on the business side is just like there's a question when a new technology comes along of like where do you apply it? Uh,
Hmm.
and this field is growing really fast, and we're right
Hmm.
at the beginning of it. And so
Hmm.
like the set of people who are thinking about all the questions of it are pretty narrow, right?
Hmm.
So yeah, I mean we've spent a bunch of time talking to clinicians in oncology and some also in cardiology and endocrinology and just trying to understand like where does it like for whom does it matter that we could turn a drug on in a certain place and off in another place,
Hmm.
or on at a time and off at a time.
Hmm.
Um, but it takes time to go through that maze. I think that's the hardest part of it science-wise. Operationally, it's like running any of these like deep techy, you know, like
Sure.
it's just already, it's like, yeah. Um, which is
Interesting. And okay. And so then what does the world look like, a few years down the road if things go really well?
Yeah. The biggest thing we're excited about is really around these remote control drugs.
Hmm.
I think that's going to be really meaningful. Um, so there's a ton of oncology drugs which uh, you can't give patients because it will kill the patient, but we know the drug will work well. Uh, and like it's really hard to engineer around these problems because tumors look almost the same as healthy tissue. So there's only a few tiny ways in which they look a little bit different in certain
Hmm.
situations. Certain tumors will be distinct from the healthy body, but usually they're not. Um, and tumors are also very heterogeneous. So if you just kill the 99% of the tumor that looks different from the rest of the body, well, guess what? You have 1% that will come back. So you can
Right.
have recurrence. So being able to go after these targets that actually are co-expressed in healthy tissue without shredding someone's liver in the process is like a really good thing to do.
Hmm.
Um, so I think that'll be really meaningful. And then on the research side, I mean, I think the bit that's been so exciting is like just getting to speed run like the optogenetics landscape, but for deep tissue. Um, so building research tools which will allow us to see in better granularity into cells to try to do forms of imaging that like we currently can't do, that might be like non-radioactive, like a radiation-free PET scan. I think a lot of that kind of stuff is also going to come out of uh,
Hmm.
of what we're doing, and that's all a little bit. Those are all sort of dreams right now. We have some ideas on how to do it, but
So
uh,
then what do you think are the biggest hurdles between here and there? Like
yeah.
science on the science side, on the engineering side, do you expect regulatory problems?
Regulatory experience. So regulatory experience means I'm just kind of taking last first on this. Uh, on the regulatory side, we need to think about like device plus drug combos. That's
Hmm.
like a complicated question.
Hmm.
Um, it's workoutable, but yeah, that's like
Hmm.
and it's a pretty easy device because it's like just like a shirt with some magnets sewn onto it, but it's still complicated. Um, on the science side, I mean, I think honestly at this point, we're just trying to make as many of these as we can against as many different proteins and targets and just try to map out the field so we know what's easy, we know what's hard.
Hmm.
And so a lot of the risk there is just like, can we find the proteins that work really well? Um,
Hmm.
we've had a lot of these funny experiences where like we looked at like a whole bunch of different proteins, like wild proteins, to see which ones were most magnetically responsive. Just to give an example, I don't know if we talked
Hmm.
about this very publicly. Um, but we did one mutation to all of these different things we thought would help, and it turned out that like immediately one of them like worked way better than all the others, and
Hmm.
so it's one of those things where like, oh, okay, like I wouldn't have uh uh like we could have kept on this one protein and just slogged it out, or we could have just switched to a different starting point and things would have gone a lot easier. So finding
Right.
the starting points is really important. Um, that's the biggest thing scientifically. There's going to be some questions I think as we go on about like uh, safety of these tools. Maybe the biggest challenge is like how do you power them?
Hmm.
So the naive answer is you can these things. So magnets aren't a power source. So you need
Hmm.
some sort of energy source to do the physical work in like a physics sense. Like work is in like the physics word for work. Um, uh, you could provide that with an LED that's shining on the proteins, but now we're back
Hmm.
to square one again because I can't shine light deep into the body. So what I need to do instead is I need to give it a chemical power source. So the way we've been doing that is we take a protein out of a firefly.
Hmm.
This is a trick that people figured out with optogenetics, where they take a firefly protein and then they take the protein that they care about, and then the firefly protein actually just glows,
Hmm.
uh, and then it glows and it hits that.
Hmm.
And you might say this seems like this light should be very, very dim, but a dim light incredibly close, like literally part of the same protein, could actually be quite bright because the
Hmm.
vast majority of photons, like from an external LED,
Hmm.
like, I think the odds of at least in some of our experiments we've done, like the odds of a photon from the lamp hitting the surface is like 1 in 10,000 photons
Hmm.
hit. So very dim, but very close is actually
I
fine.
see.
Um, so, so getting that system setting up is also like the other tricky bit to get right of like how do we self-power that, which we've got some examples that are self-powered now. But then how do we ensure that the fuel is delivered and safely delivered and all those sorts of questions. So yeah, those are things to like to figure out.
And then
Um,
how, I mean, it sounds like one question I have with anything like this is
yeah,
how close is it to applications? Actually,
yeah,
one, I've studied the history of science a little bit, and
yeah,
one interesting partial parallel, perhaps,
yeah,
was the discovery of electric light from by Francis Hawksby. So this is in 1706. The
yeah,
story is electrical attraction had been noted, and people had started to figure out that if you rub diamond in the dark, maybe you'll see some sort of light. Um, but Hawksby figures
yeah,
out that if you create a vacuum, remove all the air from a
yeah,
container, and
yeah,
you rub amber, diamond, or various substances, then you get a light that expands, and it's different than regular light. Regular light gets put out, like if
yeah,
light from a fire gets put out if you have a vacuum, but this light expanded. People saw that. They're like, okay, there's something
yeah,
new, there's something different happening here.
yeah,
But it was like then a pretty long time before you get
yeah,
to electrical applications. And
yeah,
so I'm, I guess I'm wondering to what degree, like what sort of real world application? So we've talked
yeah,
about some. It's like you want to be able to switch things on and off. So it's like, what are the real world applications? And
yeah,
should we be going after those? Like probably you're going to say yes because you are, but like how much more exploration is there to
yeah.
do? Do you see what I'm saying?
Yeah, so there's a ton more exploration to do,
Okay.
I think it is probably true that like the best versions of this are, I mean, they may look a little bit like what we're doing today, but like it may be inspired ideologically by,
Yeah.
like there's a lot of exploration to do, and these tools will keep getting a lot better. I think the biggest, so okay, so there's a thing, Thomas Edison. You're making me think on like the to continue your light analogy, right?
Sure.
He has that like maybe apocryphal quote where he's like, "Oh, I learned 99 ways to like not make a light bulb" or something like that.
Right.
Um, and I think that like that very much has been our like Edisonian, that like that style of science is how we do a lot of our stuff.
Okay.
And so if we have a good screenable measurement of the thing,
Hmm.
my experience has been like that gets better quickly.
Hmm.
If we don't have a good screenable measurement of a thing, then it's very hard to project a timeline like until it's good.
Right.
But if we're screening, we're learning a ton, and things are just getting better, even if we're not learning. And
Hmm.
like that's all great. Like you can just turn that crank, and there's a certain amount of dollars you put in, and a certain
Hmm.
amount of performance you get out, and you know what the performance number needs to be, and you can benchmark just some basic physics of like how
Hmm.
much energy would be required to switch from this state to that state, so that you know that you're in the clear if you can,
Right.
if you can corral a protein into the right state. So the question of like how far are we really comes down to like there'll be certain parts that are easy to break because like
Right.
easy to move quickly on because we have a measurement technique. Um, uh, and then on the question of like, should we do broader exploration? I think the answer is probably yes. Understanding the mechanism of these proteins is probably reasonably helpful.
Hmm.
Um, like I think it's helpful to understand how things work for sure. Um, uh, in the beginning, when we didn't know anything about these proteins, then the best thing to do was just like, well, like we could just make all of them and try all of them. And as
Sure.
long as we spend time finding ways to make that a cheap process to do, then that's what we can do. But now we're getting to the point with all this great like now it's a field. So now we
Right.
get to actually talk about like how we think these are working, which is great.
That, that
And yeah, so we'll see over the next like 2 years or 3 years whether like now things start going even faster because we have the data. Yeah.
that's really exciting. So what are the sort of range of I think real world applications here? What are the range of things? It could mean, it could just be,
Yeah.
location specific targeted antibodies. You can turn them on and off. But are there other things that you're imagining?
So uh so turning things on and off, that's a big one. You can also think about so I think about this as well a lot for like research tools and studying new things. So like if we could turn on gene expression in a given cell and then turn it off somewhere else, that would be really, really cool.
Mhm.
Just because like those are fascinating processes that like we can't usually modulate externally.
Mhm.
So something like if you think about like what New Limit has been doing,
Mhm.
with like turning TFs on and off, and you have some big advantages of that, but you also need to make sure it's active in a particular cell type or a particular place.
You said
Uh,
turning on and off TFs?
yeah, turning on and off TF like transcription factors.
Got it.
Um, so, yeah, yeah. Um, but that would be really exciting if you could turn those on and off. Um,
Interesting.
we've also made a magnetically controllable version of that like firefly protein called the luciferase.
Mhm.
Um, and then that's a really nice shortcut to make an optogenetic tool instantly mag. Because if we can have the light source flicker on and off with the magnet, and then
Right.
that's just powering an opto tool, yeah, exactly. Then you don't need to re-engineer the opto part. You just strap the second
Mhm.
controller on, and then you're good to go. Um, so drugs is probably like the single most exciting, I think, like maybe big application. Um, but you can also think a lot about like sensors is another example.
Mhm.
So uh, so the ability to um, you can read these things out really finely. That's I think one exciting thing. So if I take a video of some cells and then I apply a magnetic field on, off, on, off, on, off, I can subtract out everything that didn't like flex like that.
Mhm.
Um, and then the only signal I get back is exactly where the proteins were that I'm looking at. So
Mhm.
I can take a measurement now out of a super high background image, really messy, really noisy picture, and I can get a really clean uh, hit.
That's really
Uh,
cool.
yeah.
Huh. Interesting. So, because we're the quantum biology DAO, we have to talk about quantum for a little bit. So I mean, you're talking about like delivering power sources to the switches. I'm thinking about the radical pair mechanism and the
Yeah.
singlet and triplet for people who have not encountered that. Essentially, you have particles have spin states. Those are sort of like rotational states in some way. But the key thing in a bunch of chemical and biological contexts is that you have to have electrons having the right spin states in order to be able to join atoms together. And that's the creation of a chemical bond. And so
Yeah.
if you bring in a magnet, it changes the spin states, and changing the spin states can make it so that it's easier or harder for the chemical bonds to get created. And
Yeah.
so listening to what you're saying, I'm just thinking, well, can you apply a magnet in a targeted area and change the spin states directly? It doesn't use maglev 2, so maybe I don't
Yeah.
know, right? But can you just
Yeah.
apply a magnetic field, change the spin states of the electrons, and have it be that things don't decay as quickly? And so you have some sort of agent that's in the body. The magnetic field keeps it alive for slightly longer. Um, I mean,
Yeah.
that's, yeah. And
Yeah.
then And then I know that's not the exact thing that you're doing, but it just seems like you're saying that magnetism is this like untapped resource that people haven't looked at. It's like light has these limitations, sound has these limitations. Let's
Yeah.
use magnetism. I'm like, well, quantum is this massive unused resource. Like people don't understand it. It's almost certainly
Yeah.
doing something. And so, yeah, I
Yeah.
mean, what do you think? How do you think it does or does not bear on your work?
I think so. So look, I think now that so the longer answer is like there's all this history in the field of like people trying to get these effects and seeing these little things, and like there's all the stuff which with ferritin-based magnetogenetics and Marcus Meister's paper came in 2016 and just kind of shattered the field's hopes because they're like, like the physics numbers aren't adding up for what you're claiming. And so there's all this stuff that I think kind of like just dissuaded people from entering the field um, because of all the contention around the effects. I think now that this recent crop of work from the last 2 or 3 years is showing that like yes, there are effects. Now I think it's interesting to go back and look
Hmm.
and like now it like might be easier to have like when you have a positive control, it's always easier to do the experiment. So
Hmm.
like I think it'll be interesting to go and see these things. I would also say I think on a specific point is like just maybe like a food for thought thing is like there are excited electrons from a lot of things that are not photoexcitation.
Right.
Um, Photoexcitation happens to be a super high energy process, so it's
Hmm.
kind of it can be easy mode to pop these things up first,
Right.
but I wouldn't assume that like that's where it ends.
Hmm.
Um, and so, yeah, I think it's really interesting to like to start studying a little bit more. And yeah, um, yeah, it's just like it's really cool. Like people are, I think, I hope now people are going to go and do it.
Well, I mean, having a protein that has, a 3x response to a magnetic field is really quite convincing as far as that you can have magnetic field effects. And so,
Yeah,
yeah, maybe that'll help open the door, and then we can get into what are the mechanisms and so forth. So we're getting close to the top of the hour. If there is only one idea from today's conversation that you wanted people to remember, what would that be?
I think there's a couple. Oh gosh, um, uh, can I give a two-parter?
Sure.
I'm gonna dare. Okay, so for the half of the audience that might be interested in working in this area or getting involved or being proactive, just be like, hey, it's a growing field. Would love to have you. Reach out to us. We have a bunch of contacts on our website. We'd love to talk with anybody, and we love to get proteins in the hands of people and just have people work with this stuff. So the thing number one is like, welcome to the field. Would love to have you. Uh, Second thing is like if you're an audience member and are going to be like watching this field as it goes on, like stay tuned because the next 5 years I think are going to be really exciting. Like this huge door just got cracked open, and so um, yeah, it's some of the coolest stuff I've ever seen. So like I think it's going to be just like enjoy it. You're going to see some crazy stuff. Um, yeah,
That's pretty exciting. And I mean, I mean, you sort of answered the question I was going to ask. I was going to ask, what are you most excited about next? But it sounds like you think that the next few years in the field are going to be really interesting. I just,
yeah,
what you're describing just seems to me like there's such an incredible there's a bunch of unknowns, but it looks like
yeah,
there is a resource that can be harnessed in some
yeah.
way. And so I'm just sitting here thinking like, okay, well, what somebody's got to come in and, fund this
I
area in some big way and set up
yeah.
some, Apollo project that tries to, figure out how can we take advantage of magnetic fields inside biology. But yeah, no,
With
go
yeah,
ahead.
with yeah, exactly. I think with zero hype, like zero effort, every intent to not hype, I think
Mhm.
I would still say that this is like a CRISPR scale discovery
Mhm.
that just happened to the field.
Mhm.
And like, yeah, so what I would expect to see would be the next 5 years, kind of by hook or by crook, like no matter what happens, like there will be funding over the next 5 years to
Yeah.
just speed run all of opto to get mag versions of things. I think in particular verticals where there are good measurement tools, we will rapidly see not just proof of concept mag, but over 5 years, actually really good versions of mag tools.
Hmm.
Um, and then I think there's like, two or three tricky things to figure out before they get like actively applied into humans. But I think we've like, I mean, right now, the only people thinking hard on those questions seems to just be us.
Right.
We think those problems are solvable. As more people get in and start thinking about these problems with us and elsewhere in the field, I think they'll feel even more solvable.
Hmm.
Um, so yeah, I think it's gonna be fun. Um, yeah.
Well, great. Kira, is there anything you want to add?
No, that was perfect. I think we're right on the hour, so I'll make sure to keep it within 2 p.m. time thing.
Cool.
Thank you so much for joining us, Richard. I don't think we had any questions from the audience, but I'll definitely point them to your website in case someone has some kind of question for you. Thank you so much for taking time out of your day. And yeah, I'll let you know once the stuff is posted. And
Cool.
any other final thoughts, any zingers you want to throw in there?
I think I'm not. Yeah, I think I've got my zingers in. Um, but thank you for everyone who's in the audience too. It's also super great. Nice to have these folks live. So
I think, sorry, if you could have one more question, Matteo, did you
yeah,
want to
of course.
what you mentioned earlier? Um,
Oh,
I don't know if you want to come off audio or something.
origin
Uh,
of the photoreceptive molecule bit. Is that
Yes.
I
Yeah.
can I can stay for another. I can do a question if you just
Okay, a
want.
layman explanation for someone who had only heard today of the origin of the photoreceptive molecule.
And so okay, so photoreceptive molecule. I think what you're talking about is the Aslov domain that we started from. Um, the way that came about was just I think people looking at well actually it's a good question. I don't know where the Aslov came from. I think people are just looking for proteins that responded to light, and this protein responds to light in kind of an outrageous way, like it just changes structure a lot. Um, and then when people were looking into optogenetics, um, so people wanted to start making proteins that were controllable. They said, "Hey, look, we've got this protein that's shaped kind of like a horseshoe, so the ends are kind of close to each other. I bet we could just wedge this into the because proteins are like strings. I bet we can like wedge this into the string of another protein somewhere, and then when the horseshoe pops open like a mousetrap, that's going to really screw up whatever it's attached to." So people try that and it worked, and then and then people stick with it once one thing starts to work. I don't think it's necessarily true that ASLF 2 is like the greatest and only thing that can do this, and it has some properties that actually kind of are not so great. Um, but but yeah, that's kind of the origin of it. Is comes from like a little sub piece of a wheat plant, wheat protein, and I'm not quite sure what it's doing for the wheat, but it's probably helping the wheat look at light or something. So.